Once you have reactivated your cultures, you are then ready to do your experiments. And so this video is going to show you how to make a streak plate with the goal of obtaining individual colonies. So let's go ahead and put on our gloves. And like I mentioned, the purpose of a streak plate is to isolate individual colonies, because a single colony represents one single bacterial cell that fell on the plate and divided thousands of times until you get enough of those identical cells to be able to see them in the form of a colony. And so we like to work with colonies because they should be genetically identical all to each other, originating from one single cell. And so to practice this technique and see how effective you are at obtaining individual colonies, we are first going to make a mixed population of two different bacterial species. And we're going to use our M. luteus culture, as well as our Serratia marcescens culture, both of which you've reactivated. And so the first step is to take your empty culture tube. And this is where we're going to create or make our mixed culture. So I have my empty culture tube, and I'm going to set it in the rack for just a minute. And then I'm going to grab one of my sterile transfer pipette. Open it up. And I'm going to take 10 drops of the M. luteus culture and put them in to our mixed culture population. It's a good idea before you start working to loosen the lid, just so that as your hands get preoccupied, you're able to grab what you need. So I've loosened the lid. I'm going to take out my sterile transfer pipette. I don't want to touch it to anything because I don't want to introduce any bacterial contamination. And then I'm just going to suck up the bacteria. You want to make sure if the bacteria in your culture tube have settled to the bottom, you want to give it a good swirl so that they're resuspended. The culture should look cloudy when you do this. Okay, and then I'm going to loosen the lid of my culture tube, and I'm just going to put in 10 drops here. Let's see. Can you see that? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. If you accidentally get 11 or 12 drops, it's probably not that big of a deal. The approximate ratio is really what we're going for. I'm going to expel the rest of the liquid back in to my M. luteus culture. I'm going to put the lid back on that and set that down. This transfer pipette is no longer sterile. And so I have this bleach solution here, and I'm just going to suck that up into my sterile transfer pipette, and I'm just going to leave it in here for now. This is my waste. The bleach solution will kill any residual microbes that we don't really want growing outside of this experiment. Okay, so I put the lid back on our M. luteus culture because we don't need it for now anymore. And then we're also going to take our Serratia marcescens. Like I said, if it's settled, I gave mine a swirl to resuspend it. And we only need 1 drop of this culture because it grows a lot faster than the other one. And so the relative amount are going to be different to account for those differences in growth time. New culture, new transfer pipette. So we're going to open up a new one. And once again, we only want to grab by the bulb because we don't want to contaminate any piece that we're going to be using for our experiment. So we don't want to set this down once we open it. We want to be as aseptic as possible. So I'm going to take it out. I'm not going to touch it to anything. And then I'm going to get my next culture, and I've already loosened the lid. So I can go in here and I can suck up some culture. This is obviously much more than I'm going to need, but it's easier to do a drop when you have a lot. And we're going to take our culture tube where we're making our mixed population. And I only put 1 drop of the S. marcescens in our mixed culture. I'm going to expel the rest back into the culture tube. This is no longer sterile, and we don't need it anymore. So once again, I'm going to suck up some solution, expel it, and keep it in my waste solution. Put the lid back on my stock culture. And now here we have our mixed population. So this is 2 different bacterial species. And because they're different species, they're going to form colonies that look different from each other, which is a good way you can measure your streak plate technique when everything grows up. So we have our mixed population. Now we're ready to make the streak plate. And so we're going to need one of our cryptic soy agar plate. It came in the white box that looks like this. And we only need one. It's a good idea to label the plate before you do anything with it. So you can take your grease pencil, and I'm just gonna write streak plate on it. And then you also want to include the date. It's a good idea to include the date of when you inoculate something so that you can see how it grows. And actually, I see a little bit of contamination on this plate. I don't know if you can see that, but that little dot right there is actually bacterial growth that I did not introduce. So I'm gonna grab a clean plate. You do have some extras in your kit. So if you make a mistake or if you notice something growing on it, just grab a different plate, make sure it looks clear. We'll relabel this one. When you label, it's a good idea to label the agar side because the lid can rotate, whereas anything you label on the agar side will stay. The next thing you're gonna need are some of your sterile inoculating loops, the blue ones. We'll go ahead and open these up. And then once you take your packet, these are the loops that you're going to use. This is the area that you want to stay sterile. You want to open from this side, and this is the side that you'll be handling. You only want the loop side to actually touch the bacteria. And for our streak plate, we are going to use 4 different loops as we go. So I'm going to go ahead and open this up from the handle side. And I'm only going to grab 1 loop. And I'm going to take our mixed population, and then I'm going to dip my loop into the culture. Okay, when you take it out, you should see that you still have liquid in the circle of the loop. That liquid contains your bacterial populations. And then you're going to touch it to the agar and just go back and forth in 1 quadrant of the plate. Once you're done with this, so then now we've streaked 1 quadrant. Now we want to put this in our waste. We'll dispose of it later. Now at this point, we've taken bacteria from our liquid culture and put it on the plate. You don't want to introduce any more bacteria onto the plate. The goal is at this point now to just take the bacteria that we put on the plate and dilute it out in a stepwise manner. So make sure you see where you initially streaked. Get a new loop, grabbing from the handle side. So this is sterile once again. Go back to your plate, and my initial streak was right here. And so I'm going to overlap once, overlap twice, and then I'm gonna streak it around, but I'm not gonna overlap any more in my initial streaked area. If you overlap into the previous quadrant too much, you're gonna grab too much bacteria, pull over too much bacteria, and you won't dilute the cells out enough to be able to isolate individual colonies. So we've done 2 quadrants now, so we have to do 2 more. Once again, no new bacteria. I'm going to take my plate, I'm gonna leave the lid facing up, and I'm just going to overlap once, overlap twice, and then spread that around. And when I'm spreading it around, I'm spreading around the bacteria that I dragged over from the previous content. Once again, this is now no longer sterile and we don't need it, so we'll dispose of it. And then we do our last quadrant. So we overlap into the previous quadrant 1 time, 2 times, and then spread it around from there. And now our streak plate is complete. There's a couple of different techniques that you'll see even in this lab manual on different videos. There's not one right way necessarily to perform a streak plate. The goal is to take that initial streak and do a stepwise dilution from there, ultimately to help you end up with individual colonies. So at this point, your plate, you can't see any bacteria on it. It takes a couple of days. You can store this in a plastic bag or somewhere safe away from pets and little ones. If you put it in the fridge, it won't grow. So keep it at room temperature or somewhere warmer. if possible, and it will grow up in a few days so that you can then analyze your result.